I've combined and summarized the content from my biopsychology videos into this biopsychology revision video. If you don't understand any of the content I cover here, go to my longer videos for a full explanation. But if you just need a reminder of all the key points quickly, this is a video for you. But don't just use this video. I have a Psych Boost app, and it's designed to test your knowledge of all the topics in A-level psychology actively using flashcards. It's on iOS and Android, and you can use it for all of Paper 1 for free. If instead you want tutorial support videos with questions from all three papers, you can access over 16 hours of these, as well as hundreds of printable resources over on my Patreon. But enough of that, let's get started. The divisions of the nervous system. The human nervous system. Body-wide system of nerve cells that collects information from the world, processes this information, and then takes action by directing bodily organs and muscles via the transmission of electrochemical messages. Central nervous system, CNS, involves complex processing, includes the brain, for all conscious and most unconscious processing, and the spinal cord, which receives and transmits information and some reflex processing. Peripheral nervous system, PNS, a body-wide network of messenger neurons. Sensory, afferent neurons, take information to the CNS, and motor, afferent neurons, take information away from the CNS. Autonomic nervous system, ANS, the part of the PNS that controls actions of internal glands and is an involuntary system, so not under conscious control. Somatic nervous system, SNS, the part of the PNS that controls skeletal muscles. It's a voluntary system, so under conscious control. Sympathetic system, part of the ANS, increases bodily activities. Release of noradrenaline, activates in fight or flight response, increases heart, sweat, breathing rate, and dilates pupils. Parasympathetic system, part of the ANS, decreases bodily activities, releases acetylcholine, activates in rest and digest, decreases heart, sweat, and breathing rates, constricts pupils. Usually homeostasis is maintained. This means our internal environment is regulated by a balance between the sympathetic and parasympathetic systems. The endocrine system is a collection of glands around the body that regulate bodily functions, growth, and psychological factors. It acts by releasing chemical messengers called hormones into the blood. Examples of glands: a hormone the gland produces and the effect of the hormone. A. Pituitary gland, also known as the master gland. Hormone ACTH controls the release of hormones from other glands. B. Hypothalamus, hormone CRH, links the nervous system to the endocrine system. C. Pineal gland, hormone melatonin, modulates sleep pattern, keeping the body to a day-night rhythm. D. Thyroid gland, hormone thyroxine, modulates metabolism, the rate of energy use in the body. E. Thymus gland, hormone thymosin, stimulates the development of T cells in the immune system. F. Pancreas, hormone insulin, regulates blood sugar levels. G. Adrenal glands, hormone adrenaline, regulates the effect of the fight or flight response. H. Ovaries, hormone estrogen, develops secondary sexual characteristics in females. I. Testicles, hormone testosterone, leads to the development of secondary sexual characteristics in males. The structure and function of sensory, relay, and motor neurons. Together, they're termed the reflex arc. Cell 1 is a sensory neuron. Its function is to detect sensations, for example pain, at sensory receptors. An action potential then travels across the nerve, passing along the myelinated axon, and then the electrical signal is converted into a chemical signal to cross the synapse. Cell 2 is a relay neuron. Its function is after synaptic transmission, a new action potential forms in the dendrites. This neuron is in the spine and sends a signal to the CNS, but also immediately sends a signal along its axon to the motor neuron. Cell 3 is the motor neuron. Its function is to detect the signal from the relay neuron via synaptic transmission, and then pass the signal along its own myelinated axon to stimulate an effector, for example a muscle group in the arm moving it away from the source of pain. The process of synaptic transmission. The synapse, also called an axon terminal, this structure is found at the end of a nerve cell, a neuron, and allows neurons to communicate by passing on chemical signals. This process is called synaptic transmission. Neurotransmitters are the chemical messages released by neurons. These are either excitatory, so stimulate, making more likely, or inhibitory, making less likely, the development of an action potential, an electrical message, in the other postsynaptic neuron. The process. The action potential travels down the axon of the presynaptic neuron. This forces vesicles containing neurotransmitters to merge with the cell membrane and release neurotransmitter into the synaptic cleft. Receptors on the postsynaptic neuron's dendrite membrane detects the presence of neurotransmitters, changing the chemistry within the postsynaptic neuron. If the charge inside the postsynaptic neuron passes a threshold, a new action potential forms and the message is passed on. The neurotransmitters detach from the receptors and return to the presynaptic cell via transport proteins. This process is called reuptake. Excitation. Excitatory neurotransmitters increase the likelihood of a new action potential forming in the postsynaptic cell. When detected by receptors, the electrical charge inside becomes more positive and likely to fire. This is depolarization. Inhibition. Inhibitory neurotransmitters decrease the likelihood of a new action potential forming in the postsynaptic cell. When detected by receptors, the electrical charge inside becomes more negative and less likely to fire. Hyperpolarization. Summation is the combined effect of all inhibitory and excitatory influences, resulting in a new action potential forming or not. Unidirectional. Information can only be passed between the pre- and postsynaptic neurons in one direction. This is due to the structure of the synapse, such as the location of neurotransmitters in the presynaptic cell and receptors in the postsynaptic cell. Fight-or-flight response. The fight-or-flight response is an evolutionary survival mechanism in response to a threat. It primes the body and mind for extreme action, such as fighting for our life or escaping a threat. The body returns to homeostasis after the threat is passed. In the fight-or-flight response, a stressor is first detected by the hypothalamus. The HPA axis in the endocrine system is activated, and the pituitary gland releases the hormone ACTH. This is detected by the adrenal cortex, releasing cortisol. The hypothalamus also activates the sympathetic branch of the ANS, and the adrenal medulla is triggered via the sympathetic adrenomedullary pathway, releasing adrenaline. Role of adrenaline. Psychological effects of adrenaline include increased anxiety, attention, and alertness. The physical effects of adrenaline include increased blood flow to the brain and skeletal muscles, quick thinking and reactions, and decreased blood flow to the skin and the digestive and immune systems. Dilated pupils, improved vision, and faster breathing rate, increased oxygen. The fight-or-flight response is not designed for the modern world. It's maladaptive in most situations. It's frequently triggered by stimuli that cannot be run away from or for example, exams. In the short term, this results in acute stress, a response to immediate pressures, exciting in small amounts and giving you focus and energy, but exhausting if maintained. Long-term frequent triggering of the fight-or-flight response results in chronic stress, which can result in stress-related illness affecting the immune and circulatory systems. Hey there, as you're still watching, I'm guessing you'll find this video useful. As I release content right up to the exams, don't forget to subscribe so you know when new videos are uploaded. Also, as this video is being released, I'm on around 50,000 subscribers, and I'd love to get to 100k at some point in the next few years. Localization of function in the brain and hemispheric lateralisation. Localization of function. Functions such as movement, speech or language, and memory are performed in distinct regions of the brain. They are localized. The opposite view is the brain acts holistically to perform functions. Contralateral. Each hemisphere of the brain, right or left, controls the opposite, contralateral side of the body, including both motor and sensory pathways, and vision of the contralateral visual field. Hemispheric lateralization. Each hemisphere of the brain, right or left, is specialized to perform different functions. Language centers are in the left hemisphere. Visuospatial tasks are best performed by the right. Motor, somatosensory, visual, and auditory cortices are on both sides of the brain. Broca and Wernicke's area are only in the left hemisphere. Function of the motor cortex. Voluntary muscle motor movements across the body. It's contralateral, it's at the back of the frontal lobe and separated from the somatosensory cortex by a fold called the central sulcus. Damage. Injuring the motor cortex results in the loss of muscle function, or after severe trauma, paralysis on the opposite side of the body. Function of the somatosensory cortex. Receives sense impressions from around the body. It's contralateral. It's at the front of the parietal lobe and separated from the motor cortex by a fold called the central sulcus. Damage. Loss of sensation in the opposite side of the damage, ignoring areas of the body called neglect syndrome. Function of Broca's area. Located in the left frontal lobe only, Broca's area is responsible for speech production. Damage results in motor aphasia, difficulty producing fluent speech. Function of Wernicke's area. Located in the left frontal lobe only, it's responsible for speech comprehension. Damage results in sensory aphasia, difficulty understanding speech. Function of the auditory cortex. Receives and processes sound information from ears. Located in both hemispheres. Function of the visual cortex, occipital lobe. Visual processing in each hemisphere receives information from the opposite visual field. Clinical case study research demonstrates the loss of certain functions if damage is caused to particular areas of the brain. For example, Broca and Wernicke's case studies, aphasia, and Clive Waring, amnesia. This suggests these functions, like language and memory recollection, are localized and located in the areas damaged. The use of case studies are seen as unscientific, especially in brain research, with damage often covering multiple regions, as seen when modern MRI scans were made of Tan's brain. Modern brain scanning techniques like fMRI support older research on language centers, showing activation in the regions previously associated when healthy participants perform language tasks. Motor and somatosensory functions are highly localized. However, some systems are more distributed, and some functions like consciousness appear to not be localized at all. This suggests the correct approach in arguing for the localized or holistic nature of the brain is dependent on the function. Also, due to the high connectivity of the brain, no one area is truly independent. Split brain research. The brain is contralateral. Each hemisphere of the brain, right and left, controls the opposite, contralateral, side of the body. The visual cortex processes the visual information from the opposite visual field. The two hemispheres do communicate. They're connected by the corpus callosum, a thick bundle of 200 to 300 million nerve fibers. Cutting the corpus callosum was a surgical procedure for epilepsy, isolating each hemisphere from the other. The corpus callosotomy reduced epileptic symptoms and has few side effects for such an invasive procedure. However, unusual behavior and a sense of a loss of agency, a feeling of control, led to research. Sperry's 1968 split brain research was a quasi-experiment using 11 patients who had all undergone corpus callosotomy. Sperry projected information to each visual field, so controlling the information that each hemisphere had access to. In a series of tests, participants would be required to say either what they had seen, draw the object, or select from a number of objects that were hidden from view with their hands. Findings. Information presented to the left hemisphere could be spoken, but not spoken if delivered to the right hemisphere. However, the right hemisphere could draw or select the object by using the left hand, suggesting the hemispheres are both capable of acting independently, memories and awareness, also supporting the theory that language centers are in the left side of the brain. Gazinga's 1983 split brain research also used patients who had undergone corpus callosotomy, found that when each hemisphere of the split brain patients were presented with faces, the right hemisphere was much more able to recognize them, suggesting the right hemisphere is specialized for facial recognition. The sample of the split brain patients in these studies is small. Also, participants were compared against non-epileptics, had varying amounts of connection cut, and all undergone drug therapy. The control group used by Sperry were healthy with no history of fits. This suggests that some of the results may be due to participant variables, making the results ungeneralizable to the wider population. The experimental procedure used in split brain patients is also unlike how these individuals would process information and act in normal day-to-day life. The tasks lacked mundane realism. In reality, these people led mostly normal lives, moving their head from side to side and using other cues to pass on information to both hemispheres, resulting in low external validity. Research on split brain patients has had a fundamental impact on the psychological and philosophical understanding of both the unity of consciousness and identity, suggesting that the brain is a combination of separate intelligent processes working together. Plasticity and functional recovery. Plasticity. The brain adapts in both its function and structure as a result of a change in the environment. These changes could be due to damage, or to meet the cognitive demands of learning new skills. 4 reasons for plasticity are: learning new skills, a result of developmental change, response to direct trauma to areas of the brain, response to indirect effects of damage such as brain swelling or bleeding from a stroke. Functional recovery. The functions that were performed by areas of the brain that are now lost, neuronal cell death, or damaged are performed by undamaged areas of the brain. Functional reorganization. Synaptic pruning. Synapses that are used frequently become stronger over time. However, unused synaptic connections are lost. This makes the brain a more efficient communication system over time. Axonal sprouting is when existing neurons grow new axons to connect to adjacent neurons. Neurogeneration is the growth of new neuronal cells. Denervation supersensitivity. To compensate for the loss of axons in a pathway, the remaining axons become more sensitive, more likely to fire. This can result in side effects such as pain. Factors affecting recovery. Age. Children have the best ability to recover, then young adults. Gender. Women are more able to recover from brain damage. Access to rehabilitative therapy. Focused effort results in improvement. Constraint-induced therapy is stopping patients from using coping strategies, for example, using undamaged limbs for tasks. McGuire, 2000. An MRI of 16 male taxi drivers compared to an MRI of 16 matched age and gender non-taxi driver controls. The posterior hippocampi of the London taxi drivers was found to be significantly larger than controls, suggesting the physical structure of the brain is plastic, able to reconfigure itself to better adapt to psychological demands. Danieli's case study of 14-year-old EB. At 2, EB had a hemispherectomy of the left side of his brain to remove a tumour. This removed the language centre of Broca's and Wernicke's area. Immediately after surgery, EB had lost all language ability, aphasia. However, after 2 years of recovery, EB had recovered his language ability. Research on brain plasticity and functional recovery has practical benefits. It's been useful in rehabilitative therapy, used, for example, constraint-induced therapy, stopping patients using coping strategies like body language for communication or undamaged limbs for tasks. This makes them improve via functional reorganization, helping people return to their lives and productive work, ultimately benefiting the wider economy. Research in individuals recovering lost function can help psychologists understand more about the functions of the regions of the brain that were initially damaged. Matthias, 2015. A meta-analysis demonstrated that not everyone has high levels of plasticity, finding IQ and educational background are positively correlated with better outcomes after traumatic brain injury, suggesting some individuals have a greater cognitive reserve, helping in recovery, and some people will struggle to regain function. Ways of studying the brain: functional magnetic resonance imaging, fMRI. fMRI detects blood flow in the brain. As more active areas of the brain need more blood, containing oxygenated hemoglobin with distinct magnetic properties, these active areas can be compared to low activation areas with a lower blood supply and displayed on an fMRI image. fMRI has good spatial resolution of approximately 1 mm, precisely identifying active brain regions and patterns of activation over time while participants complete experimental conditions. fMRI is non-invasive and a safe technique for experiments compared to options that use radiation like PET scanners. Poor temporal resolution, as one image is taken every few seconds, and delay in blood flow after activity. Many brain processes are too fast to study. fMRI machines are expensive to build and operate. Also, as the patient needs to be still, experiments with body movement are not possible. Electroencephalogram, EEGs. The EEG device is a collection between 22 and 34 electrodes that are attached to a cap, and then fitted carefully to the scalp with conductive gel. The readout from each electrode is the sum total of activation of the brain cortex under the electrode. This is displayed as a series of lines showing distinct patterns called brain waves. Amplitude, the size of the waves, show brain wave intensity. The frequency, the distance between each wave, shows the speed of activation. EEG has been historically important in understanding brain activity in areas such as sleep research and medical diagnosis. It's cheaper than alternatives such as fMRI and is able to be used in experiments in which the participants move. Very good temporal accuracy, measuring brain activation with a resolution measured in milliseconds. Poor spatial activity, as the pattern is the sum of a large number of neurons in the cortex under the electrode. Also cannot detect activity deep within the brain. Event-related potentials, ERPs. Event-related potentials use the same technique and equipment as EEG, but present the stimulus many times, creating a smooth curve of activation by combining the data in a process called statistical averaging. This removes background noise unrelated to stimulus. The waveforms peaks and dips show exactly when cognitive processes in the brain happen after the stimulus is presented. ERPs allow researchers to isolate and study how individual cognitive processes take place in the brain, while EEGs record general brain activity. Like EEGs, ERPs have good temporal resolution within a millisecond sampling rate, but very poor spatial resolution. Some processes cannot be studied by ERP, as they cannot be presented a large number of times with the same response. Post-mortem examinations. Brains are precisely cut after a treatment to give them a firmer texture. Unusual brains are detected, such as brains that have suffered trauma, or from individuals with mental illnesses. These brains are compared to neurotypical, healthy brains. Broca's area responsible for language production was discovered using a post-mortem on a patient called Tan, who could only say Tan. Expressive aphasia. Damage in an area of the frontal lobe was found after Tan's death. The high spatial resolution of post-mortem allows the study of microscopic brain structures down to the neuronal level. As post-mortem is not carried out in the living brain, unusual behaviour in life and damage found is correlational. However, theories are then generalised that can be tested with other experimental techniques. Circadian rhythm. A circadian rhythm is a biological rhythm that lasts around 24 hours. We have a circadian rhythm for regulating sleep and wake, the release of hormones, and varying our body temperature and blood pressure. Endogenous pacemakers, EP, are internal body clocks that keep biological processes to time. Exogenous Zeitgebers, EZ, are external cues that entrain internal body clocks, altering EPs, to match the environment. The sleep-wake cycle is the pattern of sleep and wakefulness that repeats every 24 hours, like light and social cues in situations like jet lag. The EP will be entrained by the EZs to the new time zone, but it can take several days. The suprachiasmatic nucleus, SCN, is the EP for the sleep-wake cycle, known as the master clock. The SCN is part of the hypothalamus within the limbic system. It's located in the optic chiasm, the point in the optic nerves where they cross. When light is detected by the SCN, it sends a signal to the pineal gland, stopping the production of melatonin, a hormone that results in a sleep response. Light is thought to be the primary EZ in the sleep-wake cycle, but there are others, for example social cues, like when we time our meals or exercise, when others go to bed, or environmental cues like clocks. Sephra lived in a cave for 179 days, 6 months. The cave had no natural light or other potential extraneous variables like clocks or social cues. Sleeper's body clock, EP, maintained a regular cycle of around 25 hours without EZs, longer than the expected 24-hour cycle, suggesting the EP for the sleep-wake cycle is free-running, but needs entrainment to keep to the 24-hour day-night cycle. Psychologists have criticized the use of artificial lights in isolation experiments on the sleep-wake cycle, suggesting the artificial lights disrupted the free-running circadian rhythm measurements by acting as an EZ. Other studies that controlled for artificial light found the circadian rhythm was closer to 24 hours, with little drift. Vetter found 27 office workers who were exposed to strong blue light shifted the timing of their circadian rhythms to match the office lighting. This suggests artificial light is a strong EZ for the sleep-wake cycle. Ralph surgically transplanted the SCN between ordinary hamsters, 24-hour circadian rhythm, with hamsters with the tau mutation, 20-hour circadian rhythm, found the hamsters' sleep-wake circadian rhythm shifted to match their new SCN, suggesting the SCN is the EP in the sleep-wake cycle. Circadian rhythm research has practical applications, from understanding how blue light from devices disrupts sleep, how to combat the adverse effects of jet lag and shift work, and understanding the circadian blood pressure rhythm helps with timing drug treatments. Biological rhythms, infradian. An infradian rhythm is a biological rhythm that takes longer than 24 hours to complete one cycle. One example is seasonal affective disorder, SAD. This is when an emotional mood lowers in the winter and improves in the summer, and it's suspected this is due to the lack of sunlight in the winter. The menstrual cycle is a 28-day cycle, infradian rhythm, with 6 days of fertility. However, cycles can be shorter or longer, ranging from between 24 and 35 days. Biological aspects of this cycle include releasing the egg, ovulation, thickening of the womb lining, and losing the womb lining during menstruation. These biological changes are regulated by the hormones estrogen pre-ovulation and progesterone post-ovulation. The levels of these hormones act as endogenous pacemakers, keeping the biological processes to time, acting as an internal body clock for the menstrual cycle. It's thought that the pheromones of other women can act as exogenous zeitgebers, resulting in women who live together synchronizing their cycles. McClintock found 135 women who lived in the same dormitory at university appeared to synchronize their cycles with their friendship groups. This suggests the menstrual cycle and infradian rhythm synchronizes. Stern and McClintock, 20 women were given pads taken from the armpits of donor women to wipe on their top lip every day. It was found that the women would either shorten or extend their menstrual cycle to match the donor. This suggests infradian rhythm synchronization is due to the presence of pheromones, acting as EZs. While the use of pheromones as a chemical messaging system is common in animals, it's not widely accepted among scientists that humans have the ability to detect these signals. Trevathan found cohabiting lesbian couples did not synchronize, the optimal conditions for possible synchronization, countering McClintock's theories. As menstrual cycles can vary in length, women can appear to synchronize just due to their variability in their cycles, something not taken into consideration in all of the studies showing positive results. Evolutionary theories conflict. Some argue synchronization is advantageous, stopping one male impregnating all women at once, leading to genetic diversity in small human groups. Or maladaptive. If they synchronize, not all women can be fertilized by the male with the best genes, needing to compete with other women for him. Biological rhythms, ultradian. An ultradian rhythm is a biological rhythm that takes less than 24 hours to complete one cycle. The stages of sleep is one example. There are 4 sleep stages. Stages 1 to 3 are non-REM sleep, or NREM, termed N1, N2, and N3. After passing back through N2 and N1, stage 4 is REM, or rapid eye movement sleep. Most dreams occur in REM. One cycle takes about 90 minutes and repeats 4 to 5 times over the course of a night's sleep. Each cycle tends to include a larger proportion of REM sleep and a shorter duration of M3. The stages of sleep are identified by measuring distinctive brainwave patterns using an EEG, delta, theta, alpha, beta, gamma. These waves vary by frequency, how close the waves are to each other measured in waves per second or hertz, and amplitude, the size of the brainwave, voltage. The theta and delta brainwaves distinguish sleep stages. Sleep stages, N1, falling asleep sensation. Easy to wake, and there may be sudden body movements, hypnic jerks, a sensation of falling, and hypnagogic hallucinations. N2, harder to wake, the body is relaxed, heart rate and body temperature are lowered, and the eyes are still. N3 is the deeper sleep. Very difficult to wake, the body is most relaxed with heart rate at its lowest. Sleep becomes deeper from N1 to N3. You are less easily woken. There's a slowing of brainwave frequency and increased amplitude. REM is similar to wakefulness as seen on EEG, however the body is paralyzed. characterized by rapid eye movement and associated with dreaming. When woken, people report vivid dreams in this stage. The brain returns to N1 after REM. Dermot and Kleitman used EEG to record the brain activity of 33 participants during one night's sleep. Brainwaves followed a cyclic activation pattern, with bodily relaxation during slow-wave sleep and rapid eye movement during periods of high activation. This supports the theory that stages of sleep are an ultradian rhythm with distinct features at each stage. Shapiro, ultramarathon runners showed longer sleep after intense physical activity. Also, an increased proportion of slow-wave sleep, N3, supports the theory that slow-wave sleep is for the body's physical recovery. Hyder, EEG's patients recovering from drug overdose showed increased REM sleep, suggesting REM is for mental recovery. The overall pattern of sleep stages is consistent. However, there are individual differences, with newborn babies spending 80% of sleep in REM compared to 20 to 25% in adults. This suggests the stages of sleep is not a simple process, but adapts to the developmental needs of the individual. Ciesla found that the longest period of REM sleep coincides with the lowest point on the circadian body temperature cycle. This suggests that the circadian ultradian rhythms are connected and perhaps shouldn't be considered separate processes. Both processes could use the same internal body clock, EP, the suprachiasmatic nucleus, SCN. There are practical applications to understanding ultradian rhythms. Technology and devices based on understanding sleep stages have been developed. These track sleep and help individuals improve sleep. Avoiding waking in stage 4 stops people from feeling disoriented. This type of technology leads to a happier, healthier, and more economically productive population. If you want to try out the app, all the topics in paper 1 are free, and you can get it on iOS or Android. If you want to see model answers to biopsychology questions, or access my other resources, there's also Patreon. Speaking of Patreon, I do want to thank all of my patrons for their support. With the help of all of these students and teachers, I am able to teach part-time so I can work on the main mission of Psych Boost, the development of a free-to-watch and, hopefully high-quality, A-level psychology course. And a special thank you to Kat Posnick and Ahmed Rahmani for supporting at the developer level. So thanks to them, good luck with your revision, and I'll see you in the next Psych Boost video.